Acrylamide Polymerization. Don't worry, I'm not casting any spells, but I can cast gels. So, polyacrylamide gel electrophoresis, or PAGE, is a common technique that we use in biochemistry and in molecular biology. And so, we can use SDS PAGE to separate proteins. We can use native PAGE, where you don't unfold the proteins first. You can do urea PAGE, where you're doing RNA. So all these are different forms of polyacrylamide gel electrophoresis, where basically you get polyacrylamide, right? You get acrylamide and you polymerize it, so you get it to link up together, and you do it in a way that creates a mesh. So you make all these long chains of acrylamide with like cross links, and this forms a mesh. And you can use different amounts of acrylamide to get like a tighter and tighter mesh for smaller and smaller things, or bigger things. You can use less. So it's this really helpful technique because that allows you to separate molecules by size. So basically, so if you take a protein and you use SDS as the detergent and you like unfold it and you get it to slither through, and it's kind of like slithering through the mesh, and it's kind of like dropping a jump rope through a sea of basketball hoop rim. So it's going to just like, the longer things are going to get tangled up more and move slower. And this allows you to separate them and then see what's inside. Like how pure is your protein? Are there multiple things? You can do things like a Western blot where you use antibodies to see if specific proteins are there. So basically it's this really crucial technique. And so we do a lot of these. So in some labs you like buy your own gels. Or you don't buy your own. Well, you buy gels, like the lab buys gels, like pre-made. Like they come in like little plastic cassettes. So these are the cassette things. And so these ones are glass. So we make our own. We do use pre-made sometimes, like for gradients and stuff. But we make our own because we go through so many gels. Like I've run 740 something. Like literally, I'm dead serious. I keep track because I use the numbers for cross-reference. But basically we run a lot of gels, and so we make our own. And so today I want to tell you about how we make these gels and the really cool chemistry behind the polymerization, because you're actually getting the polymers to form. You're getting the acrylamide to actually make the mesh. Like you might have also used like Agarose Gel Electrophoresis, where that's where you used to separate like DNA fragments in like a slab instead of like a horizontal thing. So here you can kind of see there's this gap in here. I'll show you how we make these, but there's this gap, and then you pour the liquid in, and then it'll harden into a gel inside of here. So you get this really thin gel that you take things through. With an Agarose gel, it's more of like a slab that's like horizontal. But anyway, the Agarose gels, they're useful, but they're not as cool, because with an Agarose gel, basically what you're doing is you're taking this Agarose powder. So Agarose is the sugar, and you basically just dissolve it, and then you heat it up in the microwave, and so you have this really hot liquid, and then when it cools down, it gelifies. And so it forms this gel, but it's not actually like... The Agarose is already like long chains, so it's already polymers. You're not doing any polymerization when you make an Agarose gel. You're just kind of like getting it to fold up, kind of like knitting a scarf. But it does it on its own to make this gel. But with the polyacrylamide gels, you're actually making it. And how you do it is you take a mixture of some sort of, so we have like master mixes that we make, which have like buffer, and so like a pH stabilizing agent, and then acrylamide solution. And so you can use different amounts to get like a higher percentage for separating smaller things, or a lower percentage for separating bigger things. And don't worry, I'll go into all this more in detail. I just wanted to give you a look at what it looks like first. And so on its own, these are, so we have like a running gel and a stacking gel, because for SDS PAGE, typically, so what we do is you do like a running gel, which is where you're actually going to have the separating. So sometimes this is called like a resolving gel. So this has a tighter mesh, and typically I fill it to like this green line. And then on top of that, you have your stacking gel, and it's a bigger mesh. So bigger holes, and basically this allows everything to get to the start site at the same time. So basically, once they get to that tighter mesh, they're going to get slowed down more. But they can all like anything of any size can kind of travel easily through the stacking gel. And this allows like all the molecules to get a head start because you're gonna have these lanes. So we put in a comb, well, before the stacking gel sets. And so now this is, when you take this out, you're gonna have wells. And that's where you're gonna stick things in. And so you do the stacking gel so that things can, even if you like pipetted it so that that molecule was right on top where you pipetted it, it'll get to start at the same time as the thing you pipetted first. So that's the point of the stacking gel. And so for some types of gels, you don't use that. Like for urea page, we don't do a stacking gel. But then you have to be really careful that you pipette only at the very bottom really carefully and don't anyway. So when you make this, when you pour in the stuff, so basically you can just pipette it from like the corner slowly, or else it'll like overflow. You don't want it. And so, but basically at this point, like this is all liquidy. It's liquidy. It's not gonna polymerize on its own. We have to make it polymerize, and how we do that is we use an oxidizing agent. So we use like a solution of APS, ammonium persulfate, and then in the hood we have TEMED, T-E-M-E-D. And what these are going to do, we use it in the hood because it like smells really, really, really bad. So we do that part in the hood. But you don't want to add that until like right before you're ready to pour the gel, because once you add that, it's going to start this chain reaction. And this chain reaction is a radical reaction. Like by radical, I don't just mean like it's amazing. It is amazing, but it means that there's like a lone electron. So molecules atoms link together to form molecules by sharing pairs of electrons. They really don't like having a single electron. This is like super duper unstable because electrons love to be in pairs. So sometimes you have like a lone pair of electrons, like on an oxygen or a nitrogen, they have like a pair of electrons. That's all cool. But having a lone electron, that's a no-no. So we call those molecules radicals, and they start off chain reactions because they're going to try to steal an electron from something else. And so they go and they steal an electron from something, and then that thing is missing an electron and has a lone electron, so it's a radical. So it attacks something else, and you get these chain reactions. And when this is happening, when it's attacking these acrylamide, you get these chains. And it also has so it's acrylamide bis solution, and so it has like 29.1, so this is just a different thing, but so it has like bisacrylamide, and so it has things that'll form cross links between the different chains. And so by varying the percentage of that too is another way that you can change the amount of mesh. So you can imagine if you have more cross links, you might have a tighter mesh. But basically, so you make these long chains and these cross links, and so sometimes the radicals will like terminate, and so they'll bind to something else that has a free radical, and then they make a pair and everything's good. But for most of the time, what they're doing is this chain reaction where they're going from one thing to another. So it happens really quickly. And so you want to make sure that you have everything set up. Like you even have I put my pipette tips, get them ready so I can add the APS. Then slowly invert it, you don't want to get bubbles. Then go add the TEMED, slowly invert it so you don't get bubbles, and then bam, it's in here. And so what I do with an SDS page gel is I fill it to about this line. And then what I do is I have this ready to go too. So I keep like a pipette, like a big 1 ml pipette, with like a ml of isopropanol. And I just make a layer on top. And so basically the isopropanol, it's not soluble with the other mixture. So it's going to form a layer on top, but it's going to both keep the top from drying out and help you get like a nice clean line. So you can kind of wiggle it a little. You want to make sure though, so then you leave that while this part's drying. And so typically to see that when it's dry, what I do is I keep the excess, so I make the mix in like a falcon tube, and then I keep the excess in here, and I keep the pipette in the pipette tip in here. And then when it's hardened, you can tell because the pipette tip will get stuck in the gel, and it's fun because then you can kind of like play with it. But anyway, so then you know it's done. You can then pour off the isopropanol, do like a water wash. So it's helpful if you have like a squirt bottle. You can do like a water wash, pour it off, and then I should really try to dry it out. Just like put a paper towel or whatever, and it'll like suction out. And then you can add your stacking gel, and when you add your stacking gel, you want to have your combs ready. So we have different size combs. So we have like different numbers of wells. So we have like 15 wells and 10 wells, then we have 0.75 and 1.5. So that refers to the these cassettes. So these cassettes, that was a 0.75, this is a 1.5. And what that refers to is the width, or the thickness, not the width, I guess the thickness of the gel. So the back plate, it has these like spacer things on the edges. And the 0.75 will have a thinner spacer. And so then when you add the glass on top, you get that hole, and that's where the gel is going to be. So if you have a 0.75, it's going to be thinner. For other types of gel, so like when we do urea page, when we do like the bigger ones and stuff, you actually have to put your own spacers in. And so you just have like 2 plates, and then you can put different width spacers, and then clamp them together or tape them together depending on the gel in the system. But for these gel systems, so we have this caster, like, so we have the plates, and then we have these casters. The important thing is you want to prevent leaking, and so we have these, or those, I don't know what you call it, maybe those are clamps, I don't know, but then you have these casting welt tray things, and they have this rubber part on the bottom. So what you want to do is you want to put the short plate on front, and then these, they have this like hole, so you put it down through here. Sorry, it's hard to do this while filming. Okay. And then you clamp it down. What you want to do is go to the bottom and run your thumbs over the bottom and make sure it's even. If these plates are not even, you're probably going to leak. Even if the plates are even, you still might leak. So basically this goes down, and then this clamps on top. Sometimes these can come loose, and what can be helpful is if you actually stick like a pipette, like a P1000 tip in here. This can prevent leaking. So if you're starting out and stuff, it's often good to try just putting water in and seeing if the water leaks, and then you can pour the water out and cast your gel. So typically it takes probably somewhere between half an hour and an hour for each of the different sets to polymerize. And so you do the same type of thing for the stacking and the resolving gel. In terms of like, you add the polymerizing reagents right before you do the reaction, or else it'll harden like while you're preparing or whatever. And you want to make sure that like you add the 2 reagents, so the APS and the TMed, you want to like add them quickly after one another, but after a little shaking, so you don't get like pockets of that are polymerized and others that aren't. So you want to make sure it's mixed, but you don't want to introduce bubbles while mixing. So just like invert up and down a few times typically does the trick. For some type of gels, you really need to avoid bubbles. Like if you're doing sequencing gels, like urea gels, like those really big ones that are really thin. So in those cases, you can like degas it, where you have basically like this like filter device, and then you're just kind of like spinning it and getting out any bubbles. But other than that, another thing, so make the APS, it's good to make it fresh, keep that in the fridge. A lot of places say like to make it like that day or something, but I've used it several months old and it still works, but maybe it would work better if it were fresher. But yeah, so that's the basics, and let's look a little more at the science in detail. So yeah, in case anyone doubted that I was being serious about the 750 SDS page gels, yeah, I've gone through a lot, and I've casted a lot at the same time. So what's actually happening when we're doing this casting? So the key players that we have we'll have our, so the key players that we have are this acrylamide bis solution, ammonium persulfate, and TEMED or TEMD, NNNN tetramethyldiamine. Yeah, I usually just call it TMED, but I don't know how people normally pronounce it. And then you have these gel plates, and your combs and the casters and stuff. And so the basic idea is to take this solution and get it to polymerize. And so we're going to dilute this solution to various amounts, depending on how thick of a mesh that we want. And so we dilute it in like a buffer, so a pH stabilizer. So we have a consistent pH and everything's all cool. And then, but we're going to change the amount of the, like the meshiness. So, and in order to get any sort of meshiness, we need to use these APS and this TMED to get this oxidation radical reaction going. So the basic idea, so what is a radical reaction? So going back to the idea of atoms being, so like individual carbons, hydrogens, nitrogens, all of that's goodies. They're made up of individual units called atoms. And so these atoms have subatomic particles. There's the nucleus where you have positively charged protons and neutral neutrons. And then around that, you have this cloud of negatively charged electrons. And so the electrons is where the cool stuff happens usually. And so these electrons, so atoms can share pairs of electrons in order to form bonds. And so you can share a single pair to form a single bond, or two pairs to form a double bond. Electrons can also be as unbonded lone pairs. So like oxygen, it has these lone pairs. And so that's all normal. What's pretty unusual is a radical. And why it's unusual is because it's super unstable. Because molecules really hate having lone electrons. And so they can undergo radical chain reactions. And so that's what we're going to see. The other terms that we need to know are oxidation and reduction. So you can remember the mnemonic oil rig. Oxidation is loss of electrons, and reduction is gain of electrons. So one molecule gains electrons, and we say that it's been reduced. Another loses electrons, and we say that it's been oxidized. We can also represent the flow of electrons between molecules. So electrons can share and take and give electrons in order to get like the configuration that they want. So typically like having like a full valence shell or whatever, but regardless of whatever, they really don't want that radical. So we're going to see this chain reaction. And when we see the movement of a single electron, we draw that as like a one-headed arrow or like this push hook. And two electrons is this double-headed arrow. And so we're going to see some arrows in a minute. So with a radical chain reaction, what you have, it's like a game of electron hot potato. And so you have an initiation step where you need to create the radical. So you take something that's not radical and make it radical. And remember, radical just means it has a lone electron. It's unpaired. And then you enter the propagation phase. And so this is where a radical makes something else radical. And so the radical can then attack something, and then it can, then you have it can combine to make this bigger radical thing. Sometimes you can also have it where one thing, then this thing will be just take from another thing, and then that thing will become a radical, but they don't actually join. So there's different things that can happen. But what we're talking about with radical polymerization, here you have it like joining on, but staying with that other molecule. So then you have this bigger radical. And you can keep this going to get more and more things attached. So by polymerization, so a polymer is just something with a bunch of repeating units. And so we actually have like natural biological polymers are like DNA and RNA and protein. And so those are chains of nucleic acids or amino acids. And so those aren't all identical. They don't have, the units don't have to be identical, but they're similar units. And so they have the same sort of backbone, and they're connecting up. So in this case, we're connecting acrylamide. So our thing would be acrylamide. And so occasionally what can happen is termination, where two radicals come together and now they have a pair, and so they're happy and they stop attacking things. But as long as you have a radical, it's going to keep attacking things because it wants to get a second electron. And so you can take an acrylamide, and as we'll see, we can make an acrylamide radical. And once you have an acrylamide radical, then it can attack another acrylamide that's non-radical. So this is an acrylamide monomer. So a monomer, there's only a single one. A polymer, there's like more in a chain. And so then this can attack, and then you can keep getting this like path, like I don't want this electron, and so it attacks something to get another electron. And you can see here we have these like fishhook arrows because we have a single electron moving, but it's moving from one to the other, and then you're getting this long linear polyacrylamide. But a polyacrylamide, like just like a linear one, wouldn't be a very good gel, would it? You just have these strands, and we need to make a mesh. In order to make a mesh, we need to branch between the chains. But with acrylamide, you only have like a single point that you can like build from. With bisacrylamide, you can get cross links. So you have like these two sites that can be attacked. And since you have these two sites that can be attacked, it's like bifunctional. You can get these cross links between them, and you can get this branching. And so you can actually vary the amount of the various agents. So this is the bis, the NN-methylene bisacrylamide. So this is just abbreviated bis on those bottles. And you can see that there's various percentages of bis to acrylamide. And so since the acrylamide just has this one linkage point, you get straight chains, and then you can vary the amounts of this to get the chains. And so we can talk about different values, which is like you can vary the amount of total monomer, and we call that's a T, and the ratio of the two types, which is C, as we'll see in a minute. And this will control how meshy the SDS paste gel is going to be. So if you have, this is just a chart from migration chart from Biorad's website. But basically it's just showing, so this is a 7.5% gel, a 10%, 12%. They also have gradient gels, so like 4 to 15, 4 to 20. And you can see that, so for a lower concentration, a lower percentage gel, the bigger proteins, so this is like it's going to be better for separating these bigger proteins. So if you look at like a 12% gel, the 250 and the 150 kilodaltons, that's like a measure of molecular weight. So these are pretty squished together. You want to be able to tell them apart very well. Whereas in the 7.5%, you have a better separation of those, but you don't even see the little gel, like the 25 to 15 or whatever, it's already run off the gel at this point, so you don't see those. And so this is good for separating big things, whereas this is good for separating little things. Then you can see that when you have these gradient gels, you can kind of get a better separation of everything, like a separation of everything, but not necessarily like as good of a separation as you would get if you had a gel that was, the percentage was centered around what you wanted it to be, and like what was right for the protein that you're interested in. But these are really good if you're trying to separate proteins that, like, you have a big protein and a little protein you want to see, or if you just want to see what's there kind of, and you're not worrying as much about whether a particular protein you're trying to purify is really pure. So in that case, you want to kind of see, like, are those two bands or is that just one band? And so we call that like resolution, being able to tell bands apart. In order to get all that happening, you need to have to get that chain reaction going. So we need to make that radical. We need to make that acrylamide radical. And so, like, the way that we can do this is we use APS and TMED. So remember, the APS, we make like a solution of it. I keep it in the fridge, and then the TMED, we keep it in the hood, and that's the smelly one. It smells really like sulfury. So like fish eggy. So first we need to make that radical. So remember, anything radical can start the chain reaction going, but first you need to get that radical thing. And so we're going to get this radical things from APS, ammonium persulfate, and TMAD. And so ammonium persulfate, so it's this persulfate part that's going to be important. This ammonium is just like a counter ion. So a salt is a neutral combo of a cation, so a positively charged thing, and an anion, or negatively charged thing. And so you need like a counter ion to make the salt. So this is the salt form, but then when you dissolve it, these come apart. But anyway, it's this persulfate part that's important. And so basically it can split into radical sulfate ions if you heat it up, but that's not what we're going to do. Instead, we're going to get radicals from a redox reaction. And so in this reaction, we're going to have TMAD is actually going to, its electrons are going to attack APS. This is going to cause it to lose a sulfate ion. And this makes this thing really uncomfy. Yeah. And so what's going to happen is you actually get this like rearrangement and this movement of these single electrons. So that's what this these fishhook arrows showing the single electrons. And so when those single electrons move, this sulfate atom is going to actually leave as a radical. So it has a lone electron, and it also leaves this electron behind on this TMAD. So you get these radicals, and now you have these radicals, they can go initiate those chain reactions to make the gel. Going back to like how meshy your gel will be, we actually have like terms that we define. So we can have this like percent T is like the total amount of monomer. So it's the grams of acrylamide crust plus linker divided by the volume times 100. So the reason why it's like grams and mils and 100 or whatever, it's just the weight volume percent. So it's comparing the total, so the T monomer amounts to the amount of liquid. And then the percent C is like if it were to run into a monomer, how likely is it that that monomer is a brancher, so one of the bis, and as opposed to just one of the normal acrylamides. And so this is going to be your grams crosslinker divided by your grams acrylamide plus grams crosslinker. And so like what percentage of the acrylamide is actually the crosslinker one. So this is a weight percentage comparing crosslinkers to all monomers, so that's why it's a C. And so you can buy different amount, like different percent C's. And then you can make different amounts of T's from any of those. And so typically what you see, like when you see like an 8% gel or whatever, this is talking about the total T. But when you're making your own and stuff, you can use different percentage of C's. And I don't know what the different C percentages are for like pre-bought ones. I'm sure you can like find that information, hopefully. Yeah, and so then the idea, so we have this, we use a different percent in the stacking and the resolving gel. So I think the stacking gel is like 4%, but I don't want to say off the top of my head because I don't want to get it wrong. But basically, I don't know if you can see in this picture, but so like to here, you can see there's this line across here. So this part is the resolving or the running gel, and then this part is the stacking gel. And the basic idea with that, going back to, is that it's going to allow them to kind of like pile up before they run through.